Okay, so where we left off last class, we were talking about genome-wide association studies, and we were talking about how we set p-values again to consider where we need to find significance to find our signals above the noise, right? So here's our signal, and here's our noise. So again, if we're looking at, roughly 3 billion nucleotides of sequence, we need to figure out if we're trying to figure out where single nucleotides and their variances occur, we have to then consider um, how many variances we have versus how many we would expect to be variant within the percentage that we have, right? So again, if we're looking at say 100,000 individual SNPs in between people, 5% of that is about 50,000 um, SNPs between individuals, right? So 50,000 with a p-value of 0.05 just isn't going to cut it. So again, that's why we have this log of 10 to the minus 8.2, where we start to see that these SNPs, that these are reference sequence single nucleotide positions at different chromosomes, chromosome 1 and chromosome 6, where there's a significant variance as it relates to, in this case, African genomes, and in relationship to people who have had HIV and produce HIV at variant rates. So with these two loci of importance, it's also important to think about, okay, well, if we have variances related to infection, are there loci that make more sense than others? And not to say that we should rule out ones, but as you can see, this RS59 some odd number is only a few data point hits above the noise, and this RS 1131146 has a significant number of hits that lie beyond the noise ratio. And this actually kind of makes sense because this particular locus is in the HLA-B locus. So just a little bit of background, the HLA stands for the human leukocyte antigen, and the human leukocyte antigen helps basically produce an important molecule on immune cells called the major histocompatibility complex, or the MHC. And basically, what the MHC does is it exists on certain kinds of immune cells that we've discussed in class. These are called phagocytes. So, again, these are cells that consume foreign particles through the process of phagocytosis. It's a receptor mediated endocytosis. But the interesting thing is that when they do this, they then take parts of whatever it is that they have consumed, and they load it onto this protein complex called the MHC. And the ability to do this is housed in the various HLA loci that humans have. So the HLA, the human leukocyte antigen, helps um, play a really important role in how these phagocytes, again, macrophages, dendritic cells, so on and so forth. Um, Maybe I can. I can't. So that's okay. And so what ends up happening is, so if you have a macrophage, it finds something that's foreign, it consumes it, and then basically displays it on this MHC molecule and allows the adaptive immune cells to then be selected for and then respond to that particular kind of infection. So again, when we think about, and as I mentioned before, we don't want to necessarily just rule things in and out just because of how small or how many few hits they have. Because if there are hits associated with the gene or with the genome-wide association study, we should also consider those particular things as well. So as we see here, there are, again, this is in chromosome 1. Here is this gene here, the RS or nucleotide, which is in the RefSeq location of 597846663. and it has a high hit rate as it relates to African genomes related to the control of HIV. So when we think about control of HIV, what we're really talking about is how many viral particles or what is the viral burden of HIV, or in other words, how quickly does this disease progress in an individual? And that also goes back to viral burden, which also goes to transmissibility. Right? So all of these things, all these concepts kind of circulate between the two. And excuse me. So when we think about controllers, and this, graph comes directly from the National HIV Controller Study, which was done in 2010. We look at again the signal to noise ratio, and from like that particular standpoint, we see that again, here is our chromosome 6 allele, and it has a significant number of hits that rise above that threshold of minus 10 to the negative 8, sorry, to the minus 8.2, right? And here's chromosome 1, where we now lose that signal. There might be a little bit of signal that starts to approach that statistical significance, but that doesn't mean that it actually reaches the statistical significant point that we have established as being not random in the number of loci or the number of positions we are considering. And again, this kind of makes sense because if you have a SNP that is protective, in this case, we're thinking about protection because we're talking about controlling the infection, the ability to limit how quickly and aggressively the virus can replicate inside of a host, the better, or a particular, if a particular allele exists in the immune system, or the ability for the body to respond to an infection, like in this case, um, and again, the HLA component of this is a little complicated because the MHC molecule, both of them, there are two of them. There's an MHC class 1 and there's an MHC class 2. And because we have two of them, it's important to note that they basically they both form tetramers. So they have two proteins that kind of sit up here, and then they have two proteins that stud them into the membrane. So these are really big, really, really, really big protein complexes, and they have these sites inside of the extracellular effacing part of the protein that kind of looks like this. It kind of looks like a hot dog bun, if you were to look straight at it. And the pieces of an invaded pathogen sit inside of this hot dog bun. So it very much is like a hot dog bun, yeah, a hot dog and a hot dog bun, or something in a sandwich or a sub roll, if you will. And so it sits in these points of contact. And because it sits in these points of contact, you're talking about mostly the thing in here that sits inside this molecule is something called an epitope. Sorry, I'm using my fingers to type. So this is called an epitope. And an epitope is effectively a string of about 8 to 10 amino acids. So it's a small peptide string. And so, unsurprisingly, if there's a change in the DNA that makes this complex, that then affects how well or poorly something is loaded onto this, and this loading is directly involved in how efficient or how selective the adaptive immune system can be. So how well basically your whole immune system can react to an infection, then this kind of makes sense. The story kind of makes sense in that if this particular SNP allows for more efficient loading or more efficient um, accessibility to the adaptive immune system, or even just simply um, giving it some more variance as to the kinds of epitopes that are loaded onto the MHC molecule. Because again, like if you have a certain genetics, as you might imagine, certain amino acids that sit in this pocket may or may not allow for this foreign peptide or polypeptide amino acid string to bind. Because again, amino acids have to interact with other amino acids for that to load. So there's a good possibility that a protective SNP for HLA type mutation or an HLA gene could make what was previously a poorly loaded protein or sorry amino acid chain to being well loaded, and now you have the ability to uh, show the adaptive immune system more parts of the virus. So there's more potential targets for the adaptive immune system to target. So what are these elite controllers, right? So what are we talking about? And as I mentioned, we're talking about controlling infection. So less than half the percent of 38 million people globally are that are infected with HIV are considered HIV controllers, which means that this is extremely, extremely rare. But basically, they're defined by basically having almost undetectable levels of HIV in your bloodstream, despite having an active infection. And this is because viral load is directly related to how many copies of the viral genome can you get from a particular blood sample. And so when we look at this particular, total HIV DNA inside of peripheral blood macrophages, so this is basically, a cell type that gets infected with HIV. The elite controllers, right here we are, EC, has a very, very statistically significant lower, right? It has a much lower load of viral threshold. So 10 to the 0, right, is effectively one viral particle. So 1 times 10 to the 6 viral particles, and then some of them even get down into the negatives. So to 10 to down to 10 to the 4. Whereas those that do not have that elite controller status have significantly more HIV, somewhere in the order of 7.5 to 8. So, when we look at again, when we look at genome-wide association studies, again, we have to consider the fact that we're looking at association. We're not looking at uh, causation, right? In order to do causation, we have to go back in and actually put in or look at how these changes, right? The changes that we see or the changes that we don't see affect the thing that we're looking at. So, for example, in this particular case, if we're looking at this particular locus, right? If we're looking at the locus, let's say, um, RS11 31146, we would say that, okay, well, what is this mutation, right? Is it a transition mutation? Is it a coding mutation? Is it a non-coding mutation, right? What kind of mutation do we have first? Then we have to think about the consequence of that mutation. Does it change the amino acid? If it does, in which way? Is it conservative, not conservative? Does it change how much or how little RNA is produced, right? All the things that we just discussed last Friday. And so we have to consider what this mutation does to the gene that we're looking at. And and then basically compare the wild type, right? What is the sequence within the noise versus the sequence that is in the signal? And put this into different cells or even into different animal models and then simply ask the question, if we give this and only this, right? We control for everything else that we possibly can, and we only have this particular difference, does that result in the causation that we are looking for? Because again, these are really good for a starting point, but they're not the end all be all. And when we consider this, right, when we look at, say, African American descent versus European descent, there are different places where there are, different, kinds of variances, especially around the HLA locus. So this locus is very large, right? So the HLAs over here, there are the D locus. This has to do with another part of the MHC molecule. But overall, right, we can see some of the same mutations that we saw before, but they're all hovering around this HLA-B, and some, like, we have one here in HLA-C, but most of them exhibit between HLA-B, whereas in Europeans, basically splits the difference, right? But the interesting thing here also is that there's a lot more variance here. There's a lot more variance in the European study than there is in the African study, and this might also be, again, a part of the fitness model or the relative risk as it relates to how endemic or epidemic the HIV crisis is when all this data was taken. So again, we look at all the SNPs that are either in Europeans or Africans in their particular frequency and controllers versus non-controllers, right? So people who are what we would call wild type or progressive infection, right? And here are the transitions, right? So we have just regular transition C to T, and in European, right? Again, has a high rate of a percentage in both in Europeans, and but this locus, the SNP locus, 9264942, is very different than the one that is most frequently found in people who are of African descent. So again, still over zero point 0.5 in frequency, and again, really importantly, really good p-values. And when we consider this, the other part of this story has to do with that, something we talked about, which is the concept of complementarity in mutations. So it very well could be that even though this snippet occurs at position 9264942, it's because this allows for that increase in fitness by that particular change because of all the other components of the genetic background of, again, immune function and epitope loading on the MHC molecule. Whereas for people of African descent, their genetics are slightly different just because of, again, evolutionary pressure. We talked about, like, the development of or the frequency of the thalassemias and the frequency of sickle cell traits being much higher in people of African descent than of Western European descent. And because of the, again, so therefore we know that there are background genetic changes and again, alleles, not differences in genes, but differences in alleles, that particular genetic background is it has a better, HLA function with this particular SNP because it fits better with its genetics versus the genetics that are different in someone of Western European descent. So we think about, like, where these positions are and kind of where we see this particular mutation. Here are the mutations here. Position 97, position 67. We come over here to this chart and we can see that they're all involved in the pocket formation, right? So here's the HLA B pocket, here's the HLA C pocket. Sorry, the B pocket and the C pocket of HLA B. Sorry, just made a quick mistake. And so again, that pocket region, again, importantly, is really important for modulating and mediating the points of contact between the epitope, which again is foreign, it's coming from an infectious agent, to a part of the protein that is human. So this part here, right, the receptor itself is human and in nature, whereas the epitope is coming from an infectious agent or an etiological agent, so an infectious disease agent. So I'm gonna call it an IDA. So again, where these mutations occur it is in the place that helps define what epitopes can fit in here. And again, you can load a number of different epitopes. And I don't want to get too far in the weeds with this because we're going to do a whole thing on vaccines and the adaptive immune system and such. So for now, to know that this is a protein complex. It's made up of a tetramer. So we have four different proteins that make up this complex called the major histocompatibility complex, and that it has this binding domain, this pocket region, and this pocket region basically loads pieces, these 8 to 10 amino acid pieces of a foreign antigen or a foreign entity onto it to be displayed and expressed in order to activate the adaptive immune system. So that way there you can get the full complementarity of your immune system functioning, but only after this occurs. And so the other piece to this that I think is just important to put into context, which is that this piece that is being displayed to the adaptive immune system is the piece that the adaptive immune system finds. So let's say, we have a cell, it is showing you just, again, kind of make this kind of simple. If this is a macrophage and it's showing you the MHC molecule with a triangle, the T cell or the B cell that you're going to activate. So again, the adaptive immune system cell that you activate has a receptor that only looks for and can recognize. So it is selected for that particular triangle. And so the more pieces, right? Because these are only 8 to 10 amino acids. So if you can imagine, example, E. coli is 5,500 genes to 6,000 genes long in its genome. If every gene in if every gene is expressed inside of E. coli, every single protein from those genes could be broken down into 8 to 10 amino acid strings, right? So the number of potential epitopes you could load is infinite, right? With all the infectious agents that are out there, all the things that you come into contact with. But then the question becomes, in reality, right, which ones are compatible, right, that can actually fit in? Because again, this is about amino acid interactions, but also about size. So which ones can fit and which ones are chemically uh complementary to the amino acids of the binding pocket in order to load more pieces. And the more pieces you load, the more number of cells you can activate, and the greater your surveillance and your sentinel immunity becomes. So there's another mutation that has been described as being important for being protective against HIV, and this is the CCR5 Delta 32 mutation. So this is a deletion of the nucleotide at position 32. CCR5 is the complement receptor for complement component 5. Again, this is a piece of the puzzle that's, again, something that we'll probably get into later when we do immunology. But complement, just to put it into context, complement is a series of proteins that can bind and there are nine, not 10. There are nine components to complement. There are two of them. So we have C3 B and C5b. And these play a really important role in basically sticking inside of a pathogen. and illuminating them. So they kind of sit like this on the pathogen membrane, and white blood cells have a receptor that recognize this. Um, and so they can consume these pathogens by way of, again, receptor mediated endocytosis or phagocytosis. And so lacking CCR5 is not great from the perspective of most infectious diseases, but for HIV, it's really important because HIV enters through CCR5. So if you have cells that lack CCR5, you're protected. Again, it goes back to this whole concept of fitness and resistance, right? If you lack CCR5, you're not taking away the entire ability for your immune system to see pathogens. You have a whole other, set of mechanisms for that, but you are taking one of those particular mechanisms away by not having CCR5. And in the case of HIV, right, this is great because if HIV enters through CCR5, right, um, then you don't get it. Then the ability for HIV to get access to the cells becomes laden in their ability to get in through macropinocytosis or random mutation for a surface protein on their capsid to get entry into cells, right? So if you look at Northern European populations, this particular mutation occurs at about 10%. And people originally thought that this might be a kind of a functional callback to the fact that people in Northern Europe had a somewhat larger resistance to Yersinia pestis outbreaks. Um, and when we've done T fixation for this particular allele, and then done, kind of inverse linear regression, we date this to somewhere around 1300 AD, which is again the start of the second, or right around the start of the second pandemic. So when we look at the frequency of the alleles, right, 6 to 10%, over 10% in Northern Europe, and only about less than 6% in in the Baltics. When we look at the frequencies um, of mortality, right? Again, we have kind of the middle mortality, and then we had more mortality in places where we didn't have CCR5. However, yeah, we know that uh, this seems to have a association, right? So mortality is higher in places where the allele frequency is less. But something like CCR5, which has a much more complicated biology than I feel comfortable talking about, virtually, and maybe we'll come back to this on Wednesday, briefly, is that the function of complement is really, really important for bacterial infections. And again, we kind of know that, uh, biologically speaking. Whereas with HIV, this kind of makes sense because if the virus, which again is an obligate parasite, so it can't just do something else. If it can't get into cells, then it can't mutate to get into cells, right? Whereas Y. pestis doesn't necessarily have to infect um, immune cells like HIV does because it can infect epithelial cells, endothelial cells, and all the other types of cells that we talked about, which would indicate why, there's an association with this frequency in allele. But again, where you see less percentages, they can be very close in relative frequencies of mortality associated with plague. So that's it for this particular lecture. Um, and that does it for this. And we will just continue with the other lectures as we move on. Thanks.